Hello friends, this is Dr. Archana Kulkarni and welcome to my video on Indole production test. Friends, in this video I shall be covering principle, mechanism, biochemistry and procedure of Indole production test. Friends, in identification of bacteria after the gram staining, the most critical tests used are IMViC tests. IMViC is an acronym of a set of 4 biochemical tests which are performed individually and are basically used for the differentiation of bacteria. And these 4 tests are I for indole production test, M for methyl red test, V for Voges-Proska test, C for citrate utilization test, and small case I is used for ease in pronunciation. Friends, members of the family Enterobacteriaceae are medically important because they reside in the intestinal tract of human beings and animals, and some of them help in digestion of their host, while other pathogenic species cause different types of diseases. And examples of these bacteria are E. coli, members of the genera Staphylococcus, Salmonella, Enterobacter, Klebsiella, Proteus, Pseudomonas, Peptostreptococcus and Peptococcus. Friends, now let us see how the indole production test is carried out and what is the principle of this test in brief here. Indole production test is a qualitative test used to demonstrate the ability of certain bacteria to utilize tryptophan and produce indole which accumulates in the medium. This conversion is brought about by number of different intracellular enzymes generally referred to as tryptophanase. Indole production test is detected either by Kovacs reagent or Ehrlich's reagent which contains paradimethylaminobenzaldehyde that reacts with indole to produce a red-colored compound known as rose indole, which concentrates in the alcoholic layer, producing a red-colored ring at the top of the broth. Friends, to perform this test in the laboratory, you require 1% tryptone broth, a fresh culture of test bacteria, preferably 24-hour-old culture, and Kovacs reagent. Now let us see the composition of media and reagents. To prepare 1% tryptone broth, you will have to weigh tryptone 1 gram, NaCl 0.5 gram, distilled water 100 ml, and pH is set to 7.5 plus minus 0.2. Now to prepare Kovacs reagent 100 ml, you will have to weigh paradimethylaminobenzaldehyde 5 gram, isoamyl or amyl alcohol 75 ml, and concentrated hydrochloric acid 25 ml. To prepare Ehrlich's reagent, you will have to weigh paradimethylaminobenzaldehyde 1 gram, 95% ethanol 95 ml, and concentrated hydrochloric acid 20 ml. Friends, in microbiology labs, indole test can also be performed by using the following 3 media. And these media are sulfide indole motility medium, motility indole urease medium, motility indole ornithine medium. To study this indole production, 2 methods are usually used. One is called as tube method, other is called as spot indole test. Here I will explain you the procedure for tube method. Friends, tryptone broth is first prepared and distributed in the rimless glass test tubes 5 to 10 ml each, and sterilized all these test tubes by autoclaving at 15 pound pressure for 15 minutes at 121 degrees Celsius. The sterile tryptone broth tubes are then inoculated with the given fresh test culture aseptically and incubated at 37 degrees Celsius for 18 to 24 hours. Now after the incubation is over, you add 0.5 ml of Kovacs reagent to the broth culture. Mix well and observe for the presence or absence of cherry red color ring at the interface of the reagent and broth. If you get cherry red color ring at the top of the broth, that means indole test is positive. Friends, when you want to differentiate or identify non-fermenting gram-negative bacilli and anaerobes, then Ehrlich's reagent test is recommended. To perform this test, you add 1 ml xylene to the test culture after incubation so that the indole will be extracted in the xylene layer. And then you add 0.5 ml of Ehrlich's reagent from the side walls of the test tubes. If cherry red coloring appears at the interface of the broth and the reagent, then the indole test is considered as positive. Here you will see that Clostridium sordelli, which is an anaerobe, gives Ehrlich's test positive. Also bacteroids, some species of bacteroids, they give Ehrlich's test positive. Now we shall see the biochemistry of indole production test. Tryptone broth is rich in amino acid tryptophan. The bacterial cells that are able to produce enzyme tryptophan is hydrolyze and deaminate tryptophan into indole, pyruvate and ammonium ions as shown here in the reaction. Friends, indole is generated by reductive deamination from tryptophan via the intermediate molecule indole pyruvic acid. Tryptophan is catalyzed deamination reaction, during which the amine group of the tryptophan molecule is removed. And the final products that you get are indole, pyruvate, ammonium ions and some energy is released. And this reaction is catalyzed in the presence of coenzyme pyridoxal phosphate. Now here in this picture you will find the formation of quinoidal cherry red coloring at the top of the broth, indicating indole production test positive. Friends, the positive red color forms as a result of a series of reactions. The para-dimethylaminobenzaldehyde react with indole present in the medium to form a red rose indole dye. The isoamyl alcohol forms a complex with this rose indole dye which causes it to precipitate. The remaining alcohol and the precipitate then rise to the surface of the medium. Now we shall see which organisms gives indole test positive. Friends, E. coli, Proteus vulgaris, they give indole production test positive, whereas Klebsiella pneumoniae, Enterobacter erogens do not give indole production test positive. Friends, sometimes variable results also can occur showing presence of orange color in the test tube. And this is due to the presence of scatol. The scatol is nothing but methylindole or methylated indole, which is another possible product of tryptophan degradation. And if you get this orange color, that indicates the indole production test is negative. This indole production test is very helpful in the differentiation of species among the genera. For example, in the genus Proteus, it is helpful in differentiation of Proteus vulgaris from Proteus mirabilis. Now both the species are clinically important. Here, Proteus vulgaris gives indole production test positive, whereas Proteus mirabilis gives indole test negative. Also, in the genus Klebsiella, Klebsiella oxytoca gives indole test positive, whereas Klebsiella pneumoniae gives indole test negative. So also in Citrobacter's genus, Citrobacter kozeri is indole positive, whereas Citrobacter frondae is indole negative. So this is how indole production test is very helpful in the differentiation of various species among the genera. Friends, I hope you have understood the principle, biochemistry, mechanism and procedure of indole production test. In my next video, I shall be explaining methyl red test. Till then, thank you.